Hello again, we're here with the muscle tissue chapter, so let's begin. So here we will look at the three different types of muscle tissue in the body, mainly focusing on skeletal muscle in this chapter. So starting off here, skeletal muscle cells, also known as muscle fibers. They're long and thin. They generally do not divide, which we'll talk about again later. And under the right conditions, we can have some stem cells that will help produce new fibers. But you'll see that they are multinucleated. You can see there are some, just a second. You can see there are striations, and we'll see what creates these striations as well coming up in a little bit. Cardiac muscle is, we'll see, kind of a mix between skeletal muscle and smooth muscle. We have striations, just like in skeletal muscle, a single nucleus, like in smooth muscle. One really big thing, though, they do not regenerate. This is why heart attacks can be so devastating. But here we'll see intercalated discs are going to be separating at the same time, connecting these cells together. Smooth muscle cells, small and tapered. They do heal well, which is fantastic to hear, seeing as how we have blood vessel damage all the time. Therefore, we can create new blood vessels with regenerated smooth muscle cells. So, single nucleus, they are small and tapered. They interconnect, and we'll see some movements of those later on. Alright, so big thing to know is location and control. Location, skeletal muscle, we'll find along the skeleton, attaching to bones. Cardiac, we see only in the heart for pumping blood. Smooth muscle we find in various organs. So, like mentioned before, blood vessels. We see this a lot in the GI tract. So the stomach, small intestine, large intestine. We'll talk more about their functions later on in the GI tract with the digestive chapter. Other than that, you will see skeletal muscle is the only voluntarily controlled muscle type. The other two are involuntary. Thank goodness, because who wants to think about their heart beating all day long? Alright, so mostly speaking of the skeletal muscle here, we have three layers of covering. Epimysium will cover the entire muscle. We will see bundles of muscle cells. These are known as fascicles, and they are covered by perimysium. Endomysium will cover individual muscle cells or muscle fibers. Sarcolemma is directly under that endomysium, so it is the plasma membrane of that muscle cell. Transverse tubules, these are the sarcolemma diving deep towards the center of the muscle fiber. The sarcoplasm is the cytoplasm of the muscle cell. Myoglobin, we bind to oxygen to store oxygen in the muscles for later use. Myofibrils, we will see, are the contractile organelle of the muscle fiber. So muscle fibrils are inside, multiple are inside muscle cells. Sarcoplasmic reticulum, these are fluid-filled system membranous sacs. This holds a large amount of calcium for muscle contraction. Terminal cisterns, these are just dilated ends of those sarcoplasmic reticulum sacs. Other than that, myofilaments, these are going to be the small proteins that are going to be within the myofibrils that we will see contraction actually taking place. These are the thick and thin filaments. So here we'll see that. So over the entire muscle, we have epimysium. Over the fascicle, we have perimysium. And over an individual fiber, we have the endomysium. And then within that muscle fiber, we have multiple myofibrils. single one here. And within those myofibrils, we have the myofilaments. This is how we form a muscle cell. Multiple myoblasts fuse together. This is a reason why we have multiple nuclei found in skeletal muscle. Alright, so a fascicle here. Again, individual muscle fibers. So within those muscle fibers, here we see a myofibril, and you can see the thick and thin filament. The thick filament is going to be the darker, thick red line. Thin filament, the thin red line. And we'll see how they work together here in just a few slides. This is that membranous sac structure, the sarcoplasmic reticulum, that is going to hold all the calcium for muscle contraction. And we see some mitochondria for ATP production. Here's a nucleus, and here's a nucleus. Alright, so here is just laid out in sentence form, myofibrils, how they're formed, what we find in them. So thin filaments are the actin. Thick filaments are the myosin. So actin and myosin will be the contractile proteins that we see in the myofibrils. Here is a muscle fiber laid out flat, if you will. So sarcoplasmic reticulum, they put up here at the top. The myofibril, we have two seen here. Now, the big thing about the myofibril is where we find the sarcomeres. The sarcomeres are going to be how the thick and thin filaments are organized to actually cause a contraction. So the borders of a sarcomere are the Z-discs. Okay, sarcomeres, again, functional unit of the muscle. We repeat the thick and thin filaments overlying each other. The borders are Z-lines or Z-discs, if you will. And then the striations that we see in skeletal muscle are the alternating dark and light filaments, so I bands and A bands. So the thick and thin filaments are what create those striations, as you can see here. So here is a Z-disc to Z-disc. This constitutes one sarcomere. The M line is simply the center of the sarcomere. The H zone is the center area of the sarcomere. A band consists of the thick filament only. I band is thin filament only. Here is a little better picture to further see the sarcomere. The thick filament or myosin will have overlying strands of the thin filament or actin. This is so we can actually create a connection with the myosin head to an exposed actin binding site, which we will see. So contractile proteins, myosin and actin. Regulatory, which we will go over as well, is troponin and tropomyosin, very important in how connection is made between myosin and actin. So here you can see the myosin looks like two golf clubs twisted together. And then actin is seen here along the strands of tropomyosin. And then troponin, we will see, is key in attachment with calcium. Some structural proteins, titin, nebulin, alpha-actin, myomesin, and then dystrophin. These are for creating the stability and strength of the sarcomere. Okay, so actin, we have active sites for myosin to bind to, but they are covered by troponin and tropomyosin, which are the regulatory proteins. So tropomyosin is going to cover the actin. That means there is no contraction. So at rest, no contraction or connection between myosin and actin. Troponin can cause the tropomyosin to change shape, which will expose that active site on the actin. For this to happen, we have to have calcium to bind to troponin. So if calcium combines to troponin, this causes troponin and tropomyosin to change shape, which allows for that active binding site to be exposed, so myosin can attach and cause a contraction. This will shorten the sarcomere, allowing for contraction to happen along the entire myofibril. At the same time, these will all take place. All those myofibrils will contract at the same time within a muscle fiber, creating a chain reaction. So here's just another look at a sarcomere. Okay, so this is known as the sliding filament mechanism. Calcium to troponin, tropomyosin changes shape, exposes active binding site, contraction of myosin heads, binds to actin, forming the cross bridge, myosin head pivots, producing motion. Okay, so here is explaining how the Z-disc will move towards each other, towards that M line of the sarcomere. This will force the entire muscle to shorten, resulting in a whole muscle contraction overall. Okay, so here you can see those striations as well. Dark line, light line. Okay, so as contraction happens, those actin will move towards the M line and eventually overlapping over the M line. This will cause the H zone and I band to disappear. This simply means the myosin does not move, but the actin slides towards the center of the sarcomere and overlaps. And this is just a slide explaining that. So for this to happen, ATP must hydrolyze to ADP and a phosphate. The actin binding site will open. Myosin head will attach to the actin, releasing a phosphate, then eventually releasing the ADP, causing the myosin to pivot and move towards the center of the sarcomere. At that time, an ATP will reattach once the ADP releases, and this resets the myosin heads and causes relaxation. Alright, for all this to happen, we have to first release calcium from the sarcoplasmic reticulum. And we'll see how that starts. This is coming from the neuromuscular junction. So when a neuron creates an impulse that talks to the muscle cells, we will release calcium at that M bulb of the neuron. This will cause neurotransmitters to be released into the synaptic cleft, the space between the end bulb of the neuron and the muscle cell wall. Neurotransmitters bind to the muscle cell wall, opening sodium-gated channels. This will cause sodium to enter into the cell. This depolarizes the membrane, and calcium will release from the sarcoplasmic reticulum, and then we start the sliding filament mechanism. Okay, so this is showing that neuron. We have an impulse, calcium releases in, neurotransmitters out, sodium in, and then calcium is released from sarcoplasmic reticulum. And this is a nice picture where everything is laid out in steps for you to follow. And here is everything written down into sentences.